Hi. Inflammation is one of the body's defense mechanisms to get rid of noxious substances such as pathogens, damaged cells, or irritants. It involves immune cells, blood vessels, as well as molecular mediators. Inflammatory cells, plasma proteins, example complement, and fluid then exit the blood vessels and enter the interstitial space. Now, inflammation could either be acute or chronic. Acute inflammation is characterized by the presence of edema and neutrophils in the tissue. It arises in response to infection in order to clear the pathogen, or in response to tissue necrosis in order to clear the necrotic debris. It is an immediate response, less specific, and is very characteristic of innate immunity. Now let's look at some cardinal signs of inflammation. The main cardinal signs of inflammation or acute inflammation include redness, warmth, swelling, pain, and fever. Inflammation causes vasodilation, which results in increased blood flow to the area where the inflammation is taking place, and this is what causes the redness and warmth. The vasodilation is due to relaxation of arteriolar smooth muscle, which is mediated by histamine, prostaglandins, as well as bradykinin. As a result of increased vascular permeability at the postcapillary venules, there is leakage of fluid or exudate into the interstitial space, and hence the swelling that we see during inflammation. This leakage of exudate is mediated by histamine, which causes contraction of the pericytes at the endothelium, and also by tissue damage, which results in endothelial cell disruption. Sensory nerve endings are sensitized by bradykinin and prostaglandin E, and this causes pain that we feel during inflammation. Pyrogens, example lipopolysaccharide, from bacteria cause macrophages to release interleukin-1 and tissue necrosis factor, or TNF. Now, this increased cyclooxygenase activity in the perivascular cells of the hypothalamus. Cyclooxygenase acts on arachidonic acid in order to release prostaglandins. So the increase in prostaglandin E2 raises core temperature set point, and that is what causes fever. Now let's move on to the mediators of acute inflammation. First, let's look at the toll-like receptors, or the TLRs. Now, these are present on cells of the innate immune system, example the macrophages and the dendritic cells. TLRs are activated by pathogen-associated molecular patterns, also known as the PAMPs or the pumps. These PAMPs are commonly shared by microbes. CD14 is a co-receptor for TLR4, and it is located on macrophages. Its function is to recognize lipopolysaccharide, which is a pathogen-associated molecular pattern, or a PAMP, on the outer membrane of gram-negative bacteria. Now, TLR activation is going to result in upregulation of NF-kappa beta, or NFKB, which is a nuclear transcription factor. The function of NFKB is to activate immune response genes, which leads to the production of multiple immune mediators. TLRs are also present on cells of adaptive immunity, for example, lymphocytes, and therefore they play a very important role in the mediating of chronic inflammation. Now let's move on to talk about arachidonic acid metabolites. So basically, arachidonic acid is released from the phospholipid cell membrane by phospholipase A, which we have abbreviated here as PLA. So PLA represents phospholipase A. Now arachidonic acid is then acted upon by cyclooxygenase or 5-lipooxygenase. When it is acted upon by cyclooxygenase, it produces prostaglandins. So some of these are prostaglandin I, that is prostacyclin, as well as prostaglandin D and prostaglandin E. Now these prostaglandins mediate vasodilation and increased vascular permeability. So we are talking here specifically about prostaglandin I, prostaglandin D, and prostaglandin E. These are the main prostaglandins responsible for mediating of vasodilation and increased vascular permeability. And interestingly, prostaglandin E on its own also mediates pain and fever. Now, when acted upon by 5-lipooxygenase, which means that when the arachidonic acid is acted upon by 5-lipooxygenase, it produces leukotrienes. Leukotriene B4 or LTB4 attracts and activates neutrophils. LTC4, LTD4, and LTE4 are slow reacting substances which are involved in anaphylaxis, and they mediate vasoconstriction, bronchospasm, and increased vascular permeability. Now let's talk briefly about mast cells. So mast cells are widely distributed throughout connective tissue, and they are activated by tissue trauma, complement proteins such as C3A and C5A, as well as the cross-linking of cell surface immunoglobulin E by antigens. Now immediately the mast cells get activated, there is an immediate response. Immediate response involves the release of preformed histamine granules, which causes dilation of the arteriolar smooth muscle and leads to vasodilation, as well as an increased vascular permeability at the postcapillary venule. The immediate response is then followed by a late phase or a delayed response. Delayed response involves the production of arachidonic acid metabolites, and in this case, leukotrienes are the most relevant because they help in the maintenance of acute inflammatory process. Now we move on to the complement system. So complements are pro-inflammatory serum proteins that complement inflammation. They usually circulate as inactive precursors, and they can be activated by 3 different pathways. The first is the classic pathway, which involves the binding of C1 or complement 1 to an antibody-antigen complex, and therefore the complement is activated. This is the classic pathway. Now the alternative pathway is quite straightforward, and here the microbial products directly activate the complement. The third pathway has to do with the mannose binding lectin pathway, or the MBL pathway. And in this case, MBL or mannose binding lectin binds to mannose on the microorganisms, and this activates the complement. Now all these pathways we have just described result in the production of C3 convertase. Now C3 convertase converts C3 to C3A and C3B. C3B then helps to produce C5 convertase. C5 convertase converts C5 to C5A and C5B. Now C5B then combines with C6 to C9 in order to form the membrane attack complex, or the MAC, which causes lysis of microorganisms by creating a hole in its membrane. Now, C3A and C5A are anaphylatoxins. Therefore, they trigger the degranulation of mast cells, which results in histamine-mediated vasodilation of the arterioles and increased vascular permeability at the postcapillary venules. C5A is chemotactic for neutrophils, which means that it is a chemical attractant for neutrophils. Other chemical attractants include interleukin 8, leukotriene B4, and bacterial products. C3b is an opsonin for phagocytosis, which means that it binds to microbes and therefore makes it easy for them to be recognized and phagocytosed. Now, the membrane attack complex, or the MAC, lyses microbes by creating a hole in the cell membrane. And finally, let's look at the Hageman factor, or factor 12. So the Hageman factor, also known as the factor 12, is an active pro-inflammatory protein produced in the liver. It is activated upon exposure to subendothelial or tissue collagen. Once factor 12 is activated, it goes on to activate the coagulation and fibrinolytic systems. It also activates the complements as well as the kinin system. Kinin helps convert high molecular weight kininogen into bradykinin, and bradykinin causes vasodilation, increased vascular permeability, and it also induces pain. Thanks for watching. Please like this video and subscribe to the channel. More videos coming up. See you in the next one.